Welcome to Searching for the Question Live. My name is David Orban, and I am very glad to have all of you following the show. Before we start, I want to remind you that, yes, we are live simultaneously on Facebook and YouTube and Twitter, but I also invite you to subscribe to our YouTube channel, where you can, of course, also watch previous episodes with interesting people, hopefully saying interesting things together with me. Now, given that we are live, you can also ask questions or make comments, and we will see them together with my guest as they appear, regardless of what platform they are coming from. And if they are relevant, we will be happy to show them and answer them as they come. We also have a Discord community, and you can join that by going to davidorban.com/Discord, where we are continuing the conversation around the themes that are from time to time covered in Searching for the Question Live. You can also sign up for my newsletter. And of course, if you find the show valuable and the content that I create together with my team, I invite you to become a supporter on patreon.com/David Orban. Now, today what we are going to talk about is kind of a big leap. We will ask ourselves if it is possible to develop a vaccine for COVID-19 in just 6 months. And we will do it together with Hannu Rajaniemi. So Hannu, very welcome to the show. And let's hear, before we enter into the thick of our theme today and this huge challenge that you also described in a post, more about you, where you are from originally, which is nice and exotic, where you are today, what is your path and trajectory?
Thank you, David. I'm very happy to be here. I was born in Finland, in a small town in northern Ostrobothnia called Ylivieska. Grew up there, and then got very passionate about theoretical physics. Studied theoretical physics first in Oulu, which is a slightly bigger town north of Ylivieska. I see we are flying to Ylivieska there.
That's right. It should be. I prepared this beforehand, and if the gods of Google Earth are assisting us, we should be going there. Otherwise, we can always go manually. There you go.
Mm-hmm. Oh, excellent.
All right.
There we go. So,
how many people in Ylivieska?
Around 15,000. I think that's actually mentioned on the screen there. So not a huge town. Very peaceful, but a great place to grow up. And one of the things that shaped my path there was becoming very active in being a game master for a local tabletop role-playing group, which actually had quite a significant impact on.
Oh, you mean the ones that you do with dice and
exactly.
Physical cards, or was it Dungeons and Dragons, or was it Magic the Gathering? What was it?
We played all kinds of games, so a huge variety. I think my group had a strong focus on very storytelling-oriented games, so less about the game mechanics, but more about creating interesting characters and interesting worlds. So games like Amber or Vampire the Masquerade, or some of the cyberpunk games, and so on. But yeah, I think you can. There are still very active communities around these games, so you can. But yeah, this was a very significant one, which I think is one of those things that you very easily get into as a teenager, because vampires are
rather
these
than
cool outsiders.
Like this, you played it actually with physical presence, right? Because this is the video game, but the tabletop game is what gave you the initial thrust for a lot of storytelling.
That's right. So the way these games work is that you typically sit around the table. You have a group of players who create characters, and then you have a game master, or actually forget now what that's specifically called in Vampire the Masquerade. But you have this person who does not directly control a character, but who creates the world and sets the stage for the players to have adventures in. And then you typically meet for multi-hour sessions, as like once a week, or depending on your schedules. And it's very much plays like improvised theater. So there's no pre-prepared script or anything like that. The game master describes what's happening around the players, and then the players respond and tell what their characters are doing. And then if there's conflicts, typically there are rules to resolve those conflicts, sometimes using dice. But more and more, I think role-playing games have gone more into this direction of being quite light and simple with the rules, and just emphasizing different ways of social interaction. And I think there's actually an incredible future for this storytelling medium if we manage to combine it with augmented reality or online environments and things like that.
And I think that it is wonderful to allow the creativity and the fantasy of the game master to play such an important component in these games, because too many games require, as you said, following precise rules instead. Mm-hmm. And we enjoy them and we participate in them, but we cannot have a lot of input, while this kind of organization really maximizes the human creativity.
Absolutely. And it's actually not even just the creativity of the game master. It's not that the game master is the sort of mastermind behind everything, but it's really a very collaborative experience that emerges from the interactions between the game master and the players. And the players have to be equally creative in terms of how they get into the minds of their characters and try to express what the character might do in a given situation. And often players also go on to participate in the world building. They might come up with very elaborate backstories for the characters and so on. So yeah, I think there is something quite unique about it. And I don't think we've yet unlocked the full potential.
So you were saying that you got into this while in high school or university in Finland, mm-hmm. while also doing your studies in theoretical physics. Mm-hmm. And were you planning, because you were masochistic enough to do a full PhD, mm-hmm. And what a classic expectation is with a PhD is that you become a full-time researcher or maybe a teacher, an academic. Is that what you expected to, or you already knew that you wouldn't go on that path?
No, that was definitely my initial goal. Now my initial inspiration, I think, for studying physics was reading science fiction, and especially Jules Verne's 20,000 Leagues Under the Sea, I think, was the real trigger. And I wanted to study a subject that would let you discover new power sources and create spaceships or submarines and things like that, but then got swept away by the physics itself. And yes, my goal definitely was to become a researcher, to work on really cutting edge fields like quantum gravity, trying to resolve some of the remaining mysteries in physics, how to combine quantum mechanics with Einstein's general relativity so that we can explain some really challenging phenomena like black holes and the Big Bang. And I did end up studying string theory at the University of Edinburgh to kind of try to make a dent on the theoretical machinery needed for that. But then started to get a little frustrated with how far removed the field was from not even applications, just even experiments. And
quite famously, string theory is not even a theory. It's a set of theories where you pull out of the magic hat the theory that corresponds to any experiment that you can think of. So, of course, I don't know the specific area where you specialized, but the frustration that you felt is shared by an entire generation now, maybe 2 generations of physicists who have hoped in the 80s that quite rapidly we would arrive to the unification of physics, and that didn't happen. And now we are with a wonderful machine, the LHC, and we don't even know how to use it in order to get surprising answers. We got some confirmations, but the theories are not there, the experiments are not there. It's a mess. So you got out.
Yeah, I think there were also other reasons to get out beyond just being completely frustrated with the state of the field. There's a if your viewers want to learn more about some of the challenges that physics is facing now, Sabine Hossenfelder has a wonderful book called Lost in Math, which very well documents this crisis of modern physics, where we have become very wrapped up in complex theories that we can't experimentally test, and then the criteria for those theories.
Did you like The Trouble with Strings by
Peter Voigt?
No. The guy who is behind Loop Quantum Gravity.
Oh, Lee Smolin.
Lee Smolin, that's right. That's his book. Let me.
I don't think I've read that one actually.
So this is Lee Smolin's book, Trouble with Physics. Oops.
I see it recommends Lost in Math to you if you're looking at that book as well.
This is Lost in Math. Mm-hmm.
That's right. Yeah, I have not read the Lee Smolin book, but I think this is very much in the same spirit of challenging some of the directions that string theory ended up taking. No, I think that's I guess one reason I ended up leaving the field was that I also started to feel that I wanted to solve slightly more practical problems that would have more immediate impact, and was inspired by figures like Alan Turing and John Neumann, who were able to play both the very high-end theory game as well as then apply their talents to solving immediate challenges around them. Actually, in a very Manhattan Project-related manner, of course, which we'll get to. But so I, after my PhD, I teamed up with another string theorist, Sam Halliday, and we started what you could call a mathematical consultancy firm that then went on to solve much more applied engineering-related mathematical problems across many different fields, from some life science work to mobile networks to aerospace and data science and logistics and many other areas.
And after that, you did you go to Singularity University as well?
I did. That's right. That's right. So after I left this mathematics consultancy company, I explored briefly being a full-time writer. My science fiction writing career had started to develop at that point.
I have to tell you, that was a pretty good stint. I only read your first. I have to read the other 2, but I had a lot of fun with Quantum Thief. A lot of fun.
Thank you.
It is a wonderful romp. Putting together absolutely plausible, extreme scientific consequences, as well as complex societies as they evolve, diverge, and then get in conflict. So I really had a lot of fun. it reminded me of some of the early Vernor Vinge novels, like oh my God, not Deepness in the Sky, but the other one,
A Fire Upon the Deep.
Fire Upon the Deep. Yes, fire. It reminded me of Fire Upon the Deep. All right. So good. That was a good stint, as you say, of being a writer.
And of course, I do still continue to write, but I did felt at the time that I wanted to still do something real with science and technology, and Singularity University seemed like a good way to get a feel for what were the big problems that people were thinking about, and also to meet others who shared a passion for trying to apply technology to solving very big problems. So that was in summer 2013, and that was a very life-changing experience, I must say.
And at the time, of course, Singularity University was holding the summer program that for 10 weeks would have 80 people coming together from all over the world. Half of them had the tuition covered by Google, and half of them would either pay themselves or find a way to find sponsors to pay for the tuition, which was considerable, $30,000-$40,000, and then they would go through both some theoretical basis of technologies and accelerating change and all different areas, and then they would form teams to apply what they learned to change the world for the better. And then these teams would have the opportunity to continue and become part of what later would be the SU Lab Accelerator, or not. And it was a very interesting and intriguing model. Today, as many organizations, SU had to completely pivot, and SU now delivers online courses only, and we will see how this will develop through the next phases. And so after SU, what did you do?
Yeah, so being at SU really cemented an idea I had been thinking about, which was that I wanted to do something related to synthetic biology. Already with the previous company, we had some life science projects that had highlighted to me that biology was probably going to be the physics of 21st century in terms of the speed of progress and also its impact on society. And that definitely was confirmed by the things we learned at SU. And then the team I formed or team I was a part of at SU actually became a kernel of sort of the first incarnation of Helix Nano, which is still what I'm currently working on. So so some of that initial team moved on and is doing other things, but there is definitely a direct line from that SU period to what we're doing now. So there was a lot of exploration and learning on the way. I would say we stabilized in terms of the mission and direction around 2016 when my current co-founder Nikolai Eroshenko joined the team, where we really chose oncology as a key application area for synthetic biology, and which at that point had also become quite a personal mission for me. In 2015, I lost my mom to metastatic breast cancer, which was extremely frustrating, especially having started to learn about the state of the art, what was possible in the lab, but which wasn't yet available to patients.
And at Helix Nano, you apply AI techniques in order to improve the outcomes of what is a scientific approach for originally developing cancer vaccines, and now you're applying the same approach for attempting to develop vaccines for COVID-19. Is that correct? And how would you get into the next level of detail around what does that mean?
Absolutely. So in terms of what we do, it goes beyond just applying AI to cancer vaccines. Although we certainly draw upon some machine learning tools as well. It's really more of a design process. So what we try to do is, first of all, overcome some of the limitations of therapeutic modalities like conventional drugs. One amazing thing that has opened up in recent years through development of technologies like DNA synthesis, so actually being able to write DNA that encodes a protein that you can put into the human body, is that you can start designing genetic drugs. So what that means is that rather than putting a drug directly into the body, you put genetic code that gets the body's cells to actually make the drug for you. So any biological molecule you want to actually utilize to treat a disease, you can make this way. And
actually, tomorrow we will have Andrew Hassel as a guest, and Andrew was probably your teacher at SU when you were there, and he's one of the world's top experts in synthetic biology, specifically in DNA synthesis. So wonderful, and so Helix Nano takes advantage of the possibilities offered by synthetic biology to design specific molecules that can then be printed, can be synthesized, and then tested, right?
That's right. So it can, and those molecules can be, either things that get cells to produce drugs, or they can be things that induce an immune response against a virus or against a tumor. So basically, very sophisticated vaccines. And there you do have then this question of, if you can actually get the body to do anything, what should it do? So it becomes a design problem. And that's where tools like machine learning are very useful. Another very core part of synthetic biology is that you can also look for inspiration in nature. So nature has already solved a lot of the problems that you might want to solve. And the other part of this explosion in life sciences is that we also have an enormous amount of DNA sequencing data available. So we've read the genetic codes of viruses, of bacteria, of all kinds of other organisms. And you can actually look at this, all this data as a repository of biological parts that you can take and repurpose and apply to solve whatever problem you need to solve. One great example of that, of course, is CRISPR, or CRISPR-Cas9, which is originally a kind of a bacterial immune system for fighting against viruses that infect bacteria, and we've just taken it and applied it very broadly as this sort of gene editing tool. So there's this. So I think synthetic biology is really about looking into nature, finding a part that already does what you need to do, and then refining and optimizing it further, and maybe combining it with other parts. And I'm sure Andrew will have a view on how to think about this, but that's kind of the Helix Nano definition of synthetic biology. When
did you decide that, given the path that the company was on with the pandemic, you really needed to change direction and dedicate your energies and resources to trying to address the issues of the pandemic.
The very specific date was March 10th, which now seems like a very long time ago, which is actually the day after my birthday, so that made it also memorable. But what happened, David, was I was invited to give a keynote at a large pharmaceutical company on the future of the sector, and they had an ongoing COVID-19 vaccine program, and that then got me and Nikolai, my co-founder, to look at more broadly what was happening with SARS-CoV-2 vaccines. And we got quite worried about a couple of risks that we felt most of the programs were not really addressing. And the two areas roughly, which we can talk about more, where the possibility of the virus mutating and evolving, and also the possibility of so-called antibody-dependent enhancement, that if your immune response is not quite right, then the vaccine can actually make the infection worse. And these actually still, in our view, remain quite big risks for COVID-19 vaccine development.
The coronavirus belongs is a family of viruses that also SARS-CoV-2 is that the name? I always forget the name of the virus itself.
Right.
So the coronavirus causing the illness we call COVID-19 belongs to a large family that is also including the common cold. And
there are also some viruses,
some viruses causing the symptoms that we labeled common cold. And it is fairly worrisome that indeed we haven't had any vaccine for the common cold or those viruses, and it could be an indicator that it is hard, that it is something that either the viruses are, as you said, mutating very rapidly, or our immune system is dealing with them differently. So what you are saying is that at this conference, among the experts, you were worried about this kind of attitude, which reflects common scientific assumptions, but it is also a little bit self-defeating, or accepts consequences that shouldn't be acceptable in terms of the availability of the vaccine or the efficacy of the vaccine, right?
That's right. So so basically it felt like so obviously we need a very rapid response to this crisis, and we do need to develop a vaccine as quickly as possible. And so quite rightly, many vaccine programs were launched around the world already in February, March, and are now getting to the clinical stage. But there is a trade-off between rushing and thinking things through, and we just felt that a lot of these programs were not mitigating some of the fairly obvious risks up front. So just to start with the risk of virus evolving, so SARS-CoV-2, which is the name of the coronavirus that causes COVID-19, which is the disease, is a so-called RNA virus or a rhinovirus. And these viruses do tend to evolve. They get mutated more easily than DNA viruses. Mutations are how, or recombination events with other viruses is how we got into this mess in the first place. It obviously, we had a virus in bats, and then it recombined with a virus in another animal, probably the pangolin, and then that gave it the ability to infect humans. So the virus is probably not going to remain the same forever. It is going to evolve, and probably with vaccines, we're also going to create some evolutionary pressure for it to mutate and evolve. So some of the things we had been thinking about in the context of cancer vaccines were exactly about this problem: how to overcome evolution of the thing you're targeting, because cancer also evolves very rapidly. And one problem with cancer vaccines is that even if you successfully recruit an immune response against the cancer may evolve, and you then hide from the immune system, and you lose efficacy. We do have, as you're showing here, David, evidence that we are seeing some evolution of the coronavirus already. Now, just to not to make it too scary, the coronavirus does not seem to be evolving as rapidly as, let's say, the flu, which has a much higher mutation rate. And to date, there's only few instances of mutations that might be functionally relevant, that might actually potentially make the virus more transmissible, and we don't have definite proof of that. But there's also, of course, we are only seeing a very small sample of the viral genomes out there, because now we have many millions of cases around the world, and then, like, this data set here, for example, is just showing 4,000 viral genomes. So we don't necessarily have a full picture of what's going on. So
the reason you are referring to the flu, the classic flu, the seasonal flu, is because we have to try and build a new vaccine each year.
That's right.
That is the measure of the speed of mutation of the flu virus from one year to the next. The vaccine you had last year may not give you coverage.
Exactly. So the hemagglutinin protein in the flu virus mutates so quickly that we do need to make new flu vaccines every year, and even they of course don't actually work that well. So, but the flu is a harder target, I would say, than the coronavirus. So that's the good news. But I think we do need to worry about mutations in the coronavirus as well. And so that was the problem we set out to solve, using some of these ideas from cancer vaccines. So how could we create a more broadly protective vaccine?
So you had this epiphany on March 10, and you went back to the office and you said, everybody put down the hammers, put down, whatever tools you are using. You were hammering away on cancer vaccines. Starting tomorrow, we will be hammering away on SARS-CoV-2 vaccines instead. And how many companies are trying to do what you do, and why will you be better equipped in achieving the goal?
So there are now something like 110 COVID-19 vaccine projects around the world. So that includes both companies and academic groups, both large and small companies. I think we were quite well equipped to jump into this because of this genetic approach that we were using. We didn't really have to change anything we were doing in terms of the processes and workflows. We just needed to change the sequences that we were putting into our constructs. And so that made the pivot quite rapid. And we've also been very well practiced in this process of iteration and modifying what we're putting into our vectors very rapidly. So we're actually able to in real time take all the latest SARS-CoV-2 research and incorporate it into our vaccine designs. Now, we are probably not going to be the first SARS-CoV vaccine out there. There are many more clinically advanced vaccine candidates, but we could be a mitigation against those vaccines failing. So that's one way we see our role. So I mentioned the mutation risk. There is another risk that applies to the vast majority of the vaccine programs, which is called antibody-dependent enhancement. And with many viruses, including dengue prominently, we've seen this phenomenon where either your natural immune response or an immune response to a vaccine can actually make the infection worse. So roughly speaking, what happens when you have an immune response, your immune system makes antibodies against the virus. So there are molecules that stick to the virus in a way that ideally stop it from being able to infect your cells. But the problem is that if the immune system makes wrong kinds of antibodies that don't actually block the virus's ability to infect cells, now you have this antibody sticking out of the virus, and there are many cells in our bodies that have receptors for antibody tails, including immune cells. So now you've given the virus the ability to infect immune cells. And so this is the key problem for dengue. It was also seen with SARS, so the previous major coronavirus epidemic, or SARS-CoV-1, as it's now sometimes called, where the SARS candidate vaccines actually made the infection worse in animal models. So we never actually got a SARS vaccine out to the market. It went through safety testing, or like four candidates went through safety testing. And then when they were tested in animals that were virally challenged, they made the infections worse. Now, the scary thing is that the designs of the vast majority of SARS-CoV-2 vaccines or COVID-19 vaccines are identical to those SARS vaccines. So we have this cautionary tale from SARS, but we haven't really gone ahead and taken that on board.
Yeah. So the vaccine, of course, needs to be developed, and the current thinking is that it is worth pursuing parallel strategies, as you said, in order to hedge against the failure of a single approach. Is this what you mean when you are saying that a Manhattan Project for COVID-19 is needed, that the effort needs to be much more energetic, much more global, many more resources need to be dedicated to it than they are today?
Absolutely. So the original Manhattan Project tried to solve this giant existential problem of developing a nuclear weapon before the Germans did in a very, very tight timescale. And it succeeded by very carefully defining every technical problem that had to be solved, and then simultaneously pursuing multiple approaches to each problem. So there were several methods for enriching uranium that were tried in parallel, plus one method for enriching uranium. There were several bomb designs that were developed in parallel, and so on. So that way you can de-risk any kind of critical bottlenecks that might cause the whole project to fail. So you don't put all your eggs in one basket. So in this essay that on the screen, Nikolai and I proposed this kind of approach for developing a COVID-19 vaccine. So taking all the key areas, vaccine design, vaccine manufacturing, clinical trial design, clinical trial safety, etc., and throwing multiple technologies and actually developing multiple technologies to address each of them. And so we felt that this was a way to compress timelines without compromising on safety, to the point where we could get a vaccine out in 6 months. And there definitely would be economic incentives to do this. There's been several analyses of the global economic impact of the lockdowns, which I think the one argument is that a rational government should be willing to spend somewhere between 0.1 and 1% of their GDP on a vaccine, so that's obviously orders of magnitude more than is actually being spent. So,
but also an order of magnitude less than already what the decrease in GDP in 2020 is expected to be, because many countries are already forecasting 10% decrease of GDP between 19 and 20.
That's right. So what is, of course, encouraging in the US is that we do now have this.
That's good news. There is something encouraging in the US. That's
well, let's see about that. But one encouraging thing is that there is, of course, a major vaccine development effort called Operation Warp Speed, which is a $10 billion project that is trying to have some kind of centralized framework for running very large vaccine clinical trials, and which is aiming to have 300 million doses of a vaccine available by January 2021. So it's not quite the 6 months timeline, but it is quite an aggressive timeline. So there's some similarities to this effort and to what Nikolai and I proposed, which are centralized coordination and a common regulatory engagement framework, some DoD involvement, unified setup for clinical trials, and pursuing manufacturing capability build up and clinical trials in parallel. So that's similar. I think what is different is that this is still very much about vaccines developed by individual vertically integrated companies, rather than taking the full Manhattan approach of taking technologies out of those companies and putting them together in an optimal way. And in terms of pursuing multiple approaches in parallel, there are differences in terms of the delivery platforms. So we have Moderna doing a messenger RNA vaccine. We have the Oxford and AstraZeneca vaccine, which is an adenoviral vector, so a viral vaccine, and then and actually the J&J one is also a viral vaccine, and then there are protein-based vaccines and so on. But they're all delivering more or less the same thing. So there's not much diversity in terms of the vaccine design, and that's why I'm very worried about this comparison to SARS. Because it is the same design that caused this antibody-dependent enhancement phenomenon in SARS vaccines. So if one of those vaccines fails because of this ADE or antibody-dependent enhancement phenomenon, it's quite possible that they will all fail. So I don't think we have enough diversity amongst the leading approaches.
And what is the role of the Gates Foundation in the Operation Warp Speed itself, and the fact that they appeared to be willing to fund in parallel many different research projects? Do they have a more open mind compared to Operation Warp Speed, that some less traditional approaches they would be able to fund as well?
So I'm not necessarily privy to how Gates found the relationship between Gates and Warp Speed. My impression is that the Gates Foundation is more heavily involved in supporting the efforts of CEPI, so Center for Epidemic Preparedness, which is this international effort more focused in Europe. Gates Foundation has, of course, funded a lot of the companies, including Moderna, that are involved in warp speed. But I don't think Gates Foundation itself is directly involved in warp speed. So there is some overlap between the companies that they've supported, but I don't think they are actually formally part of this. Now, Bill Gates has stated that he would be willing to fund scale up of manufacturing for up to 7 of the top vaccine candidates in parallel, and that would be, I think that would be great. But I haven't heard what the latest news on that are. I do think warp speed does seem to be saying that they will do the same thing, that they will set up manufacturing for multiple vaccine candidates even before they are approved for use, which is, I think, the right thing to do.
And that is, of course, another and different challenge. Even assuming that a vaccine is going to be found with the right degree of safety and efficacy, it still needs to be produced in billions of doses. Are we beyond inoculating eggs, or it is still the way we do it?
So that'll depend on which kind of vaccine you're talking about. The traditional vaccines, definitely, like the attenuated or live vaccines, are produced in eggs. The current leading vaccine candidates, like the Moderna's mRNA vaccine, that would be manufactured enzymatically in a bioreactor. So that would be a biochemical process that still does need to be scaled up, and they are in the process of scaling that up. But it's, I would say, inherently a simpler process, so that should be more easily scaled. The adenoviral vector vaccines would be manufactured in mammalian cell culture. That's another manufacturing class. So that would be big bioreactors with tens of thousands or hundreds of thousands of liters of mammalian cells producing viruses. And then there are some protein subunit vaccines where you would actually make viral proteins and isolate them, and those would also be cell culture based. I think if I remember correctly, Sanofi has an insect cell based platform for producing those kinds of things. But I mean, I think I don't necessarily don't
in terms of time scale, because let's assume that you and everybody else working towards the same goal succeed, and we have a vaccine in 6 months, which is itself already optimistic. How long does it take to produce, I don't know, 1 billion doses?
Yeah, again depends on also what your final but let's
process.
Yeah, so let's do some back of the envelope calculations. So some of the mRNA, let's say it's an mRNA vaccine like Moderna's vaccine or Pfizer and BioNTech's vaccine, and let's say we're at the lower end of the dose, like 10 to 50 micrograms, let's say 10 micrograms to be really ambitious per dose. I think the current big RNA manufacturing facilities can produce something like 500 grams to a kilogram per month. Now that can be scaled up with money for sure, but so a like a kilogram of RNA, 10 micrograms, that would already be 100 million doses. So it could be a matter of months. I mean, it can be a little more complicated than that. For Moderna's vaccine, you then also need to formulate it with the right delivery material. But you could do 100 million.
Yeah, it is certainly not that we will need 10 years to do that.
No, I think with enough resources, it's definitely feasible to do it in a small number of months.
So you had your epiphany. You wrote your Manhattan Project proposal, and the whole company, as well as your investors, jumped on board.
Mm-hmm.
That's wonderful.
Yeah, I think this was the transition was so. Actually, I should give some credit also to our team, because they had already, before we made this flip, proposed a hackathon, a sort of side project on COVID-19. So we had even actually had been thinking about it a little bit, but then understanding the gaps in the vaccine landscape really made it possible to jump in. And yeah, the response from our investors certainly was universally positive and supportive, and then some of them, including Sam Altman, gave us some additional backing to make sure that we could move fast without worrying about runway or resources.
Yeah, that would have been my next question, because traditional assumption is that developing anything health related is like $1 billion, and maybe that is wrong by itself. But maybe your existing funding was something that needed to be complementing, and it needed to be complemented, and you are confirming that you received some additional funds in order to pursue what you want to do. So,
yeah, so just a quick comment on that, David. So it's not really that the early stage R&D costs billions of dollars. I mean, actually, one of the reasons why I remain very excited about synthetic biology as a field is that that you can actually do research now very cheaply with small teams, because amongst other reasons, DNA synthesis is so cheap. It's really at the clinical stage where it gets very expensive, where you have to manufacture large amounts of your drug, you have to recruit patients, you have to make sure that you treat them correctly in the clinical environment, and so on. So it's not that bad at the stage where we are at, where we are doing relatively rapid animal experiments, but certainly we'll need more resources or partners for the clinical stage.
And people who are interested can go to Helix Nano, and they can also write to Vaccine Manhattan Project at helixnano.com, and hopefully you will get more and more people corralling around an idea, or the idea that you described, or a variant of the idea that you described, because what you highlighted in terms of the dangers of an approach that is too even if there are variations, it is still not representing a full spectrum of different approaches, and as a consequence, it could fail. Is too frightening. We don't want periodic lockdowns to characterize the next decade. We want to be able to be together again as humans.
No, absolutely, and just to mention, David, what we're doing on that front. So given that we do have a big effort like Warp Speed on the sort of more mainstream side, we are actually now in the process of launching a non-profit to address some of these risk factors that we talked about in the Manhattan Project. So actually a separate entity from Helix Nano, completely with the mission to create a common good for vaccine development, to resolve some of these big scientific uncertainties that we have, especially around immune enhancement of infection, to make sure that we actually end up designing safe vaccines and are able to detect some of these risks early if they are realized. Hopefully not, but I think we can, with a relatively small effort, actually make a big difference there.
Wonderful. Hannu, thank you very much for being with us today. Congratulations for your project, and good luck with your next steps, including the nonprofit that you are launching.
Thank you very much, David.
So thank you, everybody, for being here today with Searching for the Question Live. If you speak Italian, I invite you to also join and subscribe my Italian language YouTube channel. You can find it easily on davidorban.com/YouTube Italiano. I want to thank our Patreon supporters who help me and my team produce this show as well as the others. And I will see you at the next episode of Searching for the Question Live.